How do DNA mutations occur?
A mutation in the DNA sequence of an organism can result from errors in DNA replication during cell division, exposure to mutagens, or a viral infection. Germline mutations that occur with procreation can be passed on to offspring, while somatic mutations that occur in body cells are not passed on. How does DNA and RNA work together to express and transmit genetic information?
DNA and RNA work in a two-step process, transcription and translation. DNA holds the genetic information for building and operating an organism and stays within the nucleus. RNA polymerase is an enzyme that creates a strand of nucleic acid from a single-stranded DNA template, and this new RNA strand is typically mRNA, though there are other types of RNA with different purposes. mRNA carries the instructions out of the nucleus and into the cytoplasm to the ribosome, where it is read, turned into codons, then matched with its corresponding amino acid. Why do errors during DNA replication occur?
Errors during RNA synthesis occur due to mistakes by RNA polymerase, which lacks proofreading, or from damage to the DNA template that is then incorrectly copied into RNA. Other factors include chemical damage to the RNA or DNA, environmental agents like radiation or chemicals. How do the structural differences between DNA and RNA affect the way they function?
DNA contains deoxyribose sugar, while RNA contains ribose sugar. Deoxyribose sugar lacks a hydroxyl group, unlike ribose sugar, which makes the DNA more stable and is necessary for long-term storage. RNA's hydroxyl group, extra hydroxyl group, makes it more chemically reactive, which contributes to its shorter lifespan. How does DNA cause aging?
DNA causes aging through the accumulation of damage that leads to mutations, epigenetic changes, and cellular dysfunction. This damage can result from sources like UV radiation and metabolic byproducts, and as DNA repair systems become less efficient over time, the damage accumulates. Why is DNA confined to the nucleus, but RNA can travel in and out?
DNA is large and tightly packed into the nucleus. Its size prevents it from passing through nuclear pores that RNA can enter and exit through. DNA is also a protected genetic blueprint and serves as a template to help create mRNA, which is temporary and can be recreated. Why does RNA have a different fourth nitrogenous base?
RNA uses a different fourth nitrogenous base because it has a single chain, unlike DNA, that uses two. While DNA contains deoxyribose and thymine, RNA contains ribose and uracil. Why is it safer for the cell to send out a copy of the genetic information instead of the original to the rest of the cell?
The reactive enzymes and high activity of the cytoplasm is capable of damaging DNA if it were stored outside of the nucleus, or possibly causing mutations. RNA is disposable and can be replicated, but not without DNA, therefore it must stay protected in the nucleus. What is RNA made of?
RNA is a nucleic acid present in all living cells that has a structural similarity to DNA. Unlike DNA, however, RNA is most often single-stranded. RNA is made of a chain of nucleotides, which are each composed of three parts: a ribose sugar, a phosphate group, and one of four nitrogenous bases: adenine, uracil, cytosine, or guanine. These nucleotides link together to form a single-stranded molecule with a backbone of alternating ribose sugars and phosphate groups. If a mistake is made when copying the DNA to RNA, how might the mistake change how the cell behaves?
Since the copying of DNA to RNA is a step towards creating proteins, a mistake in the transcription process could result in non-functioning or malfunctioning proteins, in which case the latter could lead to cell damage. If this dysfunctional transcription of mRNA continues, it could lead to cancer or aging or even death.